Hello, I am Professor Sims, and in this video I will discuss differential staining of endospores, capsules, and flagella. This is the fifth in the series of 10 lab sessions held as part of my online Laboratory for the Fundamentals of Microbiology course. If you're currently a student enrolled in this course, please consult the syllabus and the Moodle site for assignments, quizzes, due dates, and other course information. The learning objectives for this unit include learning how differential and structural staining techniques are performed in order to identify endospores, capsules, and flagella. We will learn about the formation, structure, function, and morphological characteristics of these sub- and extracellular structures. I wanted to note at the start here that since the Foldscope instruments we use for the online labs have limited resolution power, we will not be able to perform the stains and view these structures in a hands-on lab situation, but you will still learn about them and you still need to be able to know the steps of the staining procedures and be able to identify and describe the structures from figures and photographs. An endospore is a dormant form of a bacterium that allows for it to survive in poor environmental conditions. Spores are resistant to heat and chemicals because they have a tough cortex layer with an outer covering made of the protein keratin. Sporulation is the name of the process in which endospores are formed. Sporangium is the name given to the mother cell, which is the cell that is in the process of forming a spore. Germination is the name of the process in which endospores restart their metabolic activity and the cells become vegetative. And vegetative cells are those that are growing and dividing. They're reproducing. Essentially, sporulation is like a cell going into hibernation, while germination is like when hibernating cells wake up and start to make babies. Like bears and such do in the winter, and then they wake up and mate in the spring. It's essentially that. Except bacteria are capable of producing spores at any time where the conditions are no longer optimal. So if the pH has changed or they're low on food or water, anything like that. And bacteria are not necessarily doing these processes like all at the same time. It's not a seasonal thing. And that's why you may very well see cells at different stages of sporulation and germination all on the same slide. Even though it's a pure culture and the cells are all from the same species, they may end up looking really different if they're in different stages of sporulation or germination. For example, here is a normal vegetative cell. It has no endospores. It's actually undergoing binary fission. It's growing, it's reproducing. But during these steps here of sporulation, there is a forming endospore that's surrounded by sporangium, the mother cell. And in fact, right around here in this stage, these are the easiest ones to view, the ones that have the sporangium still intact, because, well, you'll see during the staining technique, the sporangium will be a different color from the spore, which makes them a little bit easier to view. And then the last stage here of sporulation is where you no longer have a mother cell, no sporangium is left, and all you have left is the spore. So this is essentially a hibernating cell. And when conditions become optimal again, that cell will germinate and then become a vegetative cell again. Be sure to read in the text about all of these stages and how the cortex here and the forespore and the spore coat are formed. Endospore staining is an important technique for identifying Bacillus and Clostridium. These are two genera of endospore-producing bacteria that contain clinically significant species. For example, Bacillus anthracus here. That is the bacteria that causes anthrax, respiratory anthrax, cutaneous anthrax, the skin. This has been of particular interest because of concern that its spores could be used as a bioterrorism agent. Also, you have Clostridium botulinum. He produces botulism, and Clostridium tetani, which causes tetanus. Another endospore-forming Clostridium is Clostridium difficile. It is a particularly important species because it's responsible for a hospital-acquired infection known as C. diff. It's notoriously antibiotic resistant, very difficult to treat. The procedure itself is called the Schaefer-Fulton method. This is the most commonly used endospore staining technique. And very similarly to the acid-fast stain, it uses heat to push the primary stain into the endospore. The primary stain here is malachite green, and the heat helps the malachite green to penetrate the cortex layer of the endospore. And the decolorizer in this step is water. Because malachite green is water soluble, it can be removed from the sporangium or from vegetative cells with just water. And then the malachite green will only remain if it's trapped inside of that thick cortex layer of the endospore. Safranin is the counterstain, and it stains vegetative cells and sporangia, but not the endospores. The green endospores can appear within the pink sporangium. If no endospores are present, again, then that is a vegetative cell. And sometimes you can have spores that are just by themselves with no sporangium left. Alternatively, your endospores may appear clear if the primary stain was not able to penetrate the cortex. So there's an example here where the spore is mostly clear. You can see where some of the malachite green got in there. Here's another one here. But sometimes it just can't penetrate, and in that case, the spores will be clear. Also, sometimes you can see endospores when you're doing a simple stain like this one. The endospores will be clear voids within the cell because, again, with a simple stain, the dye is not able to penetrate the endospore. Here is a quick demo of the endospore technique. I have edited this video for time, but also I wanted to cut out some parts where she says that there's no decolorizing step for the endospore stain. That is incorrect. The decolorizer for the endospore stain is water because malachite green is polar. It is water soluble, which allows the water to remove it from the vegetative cells in the sporangium. Therefore, water is the decolorizer for the endospore stain.
The endospore stain is similar to the acid-fast stain in the fact that we're going to use heat. The difference is there's only the first stain and the second stain. The primary stain in this case is not staining the outside wall of the bacteria, it's actually staining the endospores. Then your counter stain will stain the wall of the bacteria. So just as we did with the acid-fast stain, we're going to put our slides that already have a fixed culture on them that are labeled on our heat. And once again, we don't need it rapidly boiling, we just need to see steam coming off of it. So I can see the steam, the condensation forming. As we did with the acid-fast stain, we're going to drop the stain on there. Oh, the first stain in the endospore stain is called malachite green. So we're going to put our malachite green on there, making note not to get too much in the water. That is going to steam for the 5 minutes. Remember to watch it. So don't just walk away and come back 5 minutes later. You want to watch it. If it starts to dry out, you add more stain. So that's going to steam for 5 minutes. The steam allows the stain to penetrate the endospore wall. Once it penetrates the wall, it can't come out. So then when you rinse it, it remains in the wall of just the endospore. The actual cell itself will lose the stain and be colorless. Okay, so after our 5 minutes, we're going to gently take them off. We're going to rinse with water as we've done before. Now, if your slides get extremely hot, that probably means that your water was too high, but if they do, you don't rinse them right away because they're going to crack. Mine were too hot. If they're cool to the touch, you're good to go. So rinse them really good like we've already. Once again, don't be afraid to rinse, rinse, rinse. You can't really over rinse. We're going to go immediately to the counter stain, which is safranin. You saw safranin as the counter stain in the gram stain. So what we're looking at here, so after that's set its time, I'm going to rinse. So again, rinse, rinse, rinse. Don't be afraid. You can't really over rinse. Now you don't want to spray directly on the culture. And as we did before, we're going to blot dry. Okay? After we stain, we blot dry.
So I've included the link to the complete video in the description, and it is worth a watch as she explains the process in a bit more detail. Just keep in mind the correction that I made about the decolorizer. Now for experiment 1, you will practice identifying vegetative cells, sporangium, and endospores using this video link here that I made. You'll also try to identify the location of the endospores, as this characteristic is often used in the identification of Bacillus and Clostridium species. So this is a good description here of your locations. You have terminal, you have central, and you have subterminal. It can often be very difficult to distinguish the difference between subterminal and terminal. You just kind of have to try to look really, really closely to see if there's any sporangium left on the other side of the spore. That generally means that it is subterminal. Capsules are going to be the next structure we're looking at. Glycocalyx capsules are a network of polysaccharides and proteins, and they are closely associated with the cell membrane or the cell wall. Glycocalyx capsules are different from glycocalyx slime because because a slime layer can be easily washed away, whereas capsules cannot. Capsules are really quite sticky. Capsule production increases the virulence of some microbes such as Bacillus anthracus, Cryptococcus neoformanis, and several pneumonias. Capsules make the cells less vulnerable to phagocytosis. They also aid in adhesion, for example, sticking to the upper respiratory tract, to the lining of your upper respiratory system. Like pneumonias, that's why it's so hard to, you have fluid in your lungs and it's so hard to get it out. That's because of the capsules. And they also prevent cells from drying out due to their high water volume. They can protect bacteria from certain bacteriophages, viruses that attack bacteria. So they're very beneficial for the bacteria themselves, but they're not so beneficial for their host species. They're highly virulent. Capsules are composed of mucoid polysaccharides or polypeptides that repel most stains. The capsule stain technique takes advantage of this characteristic by staining around the capsule. So you have a combination of positive and negative stains in order to visualize the capsules. You use a basic stain like your crystal violet or safranin or methylene blue in order to stain the cell itself. And then you use an acidic stain to stain the background. And then the capsule itself, it doesn't have a charge, so it's not attracted to the basic dye or the acidic dye. It doesn't have a positive or a negative charge. It has a neutral charge. So if you stain the background and you stain the cell, then the only thing left that won't stain will be the capsule, and it will appear as a halo around the cells. It can be a really large capsule like this here, or you can have very small capsules that are very, very difficult to see. Here's a quick demo of the capsule stain technique. Now for experiment 2, you're going to be watching this video here. in which you will practice identifying and differentiating from the images of capsule stained slides. You're going to make sure that you can point out the bacterial cell itself, the capsule surrounding the cell, right? So you have the cell, the capsule around the cell, and then the background. It's really important that you can differentiate those three things. And remember that sometimes the capsule may be so thin that it's barely visible. Even with the highest power objective and the fanciest light microscopes, they still can be very difficult to see. But in general, you're looking for something like this. Flagella are tail-like cellular structures. They are extracellular structures. They're used for locomotion. Because they're so thin, and they are very thin, they can be less than 0.2 micrometers in diameter. And because they're so thin, they're typically unable to be seen under a light microscope. You first have to do a very specialized staining technique in order to thicken the flagella. Generally, you're going to be applying a mordant first. Sometimes the mordant has tannic acid, or it can also have potassium alum, which is a heavy metal. And then that mordant coats the flagella, and the specimen is then stained with pararosaline, most commonly, or can be stained with a basic fuchsia dye. The necessity of the tannic acids and heavy metals make this staining technique uniquely hazardous, and it should only be performed under a laminar flow hood for this reason. In fact, I don't even have my students in my on-site labs perform the flagellate stains because of the heavy metals and tannic acid that's involved. So we actually only view pre-made slides in class. Flagellate staining and characterization are routinely used in identification of suspected pathogens. Flagella aid in pathogenesis by allowing for chemotaxis, directed motility, and it can also aid in mechanosensory response, adhesion, biofilm formation, and secretion. And all of these things increase the virulence of some of these flagellate species. The location and number of flagella can be useful in classifying and identifying bacteria in a sample. The four major classifications of flagellates are monotrichous, amphitrichous, lophotrichous, and paratrichous. Monotrichous here have a single flagella at one pole, right? Single flagella at one pole. Amphitrichous have a single flagella at each pole. So one here, one here. Amphitrichous, I like to remember amphitrichous. If you know what ambidextrous means you can write and do things with both hands with equal ability. So just think about ambi, amphi, one at each pole, right? Left hand, right hand. Lophotrichous is when it has two or more flagella and it's either at both poles or at one pole. So lophotrichous, I kind of remember that by saying that they're lopsided. They always have more than one flagella at a pole, but it's lopsided. Sometimes it's both poles and sometimes it's only one pole. And then peritrichous, I remember peritrichous by the fact that it has flagella all around the perimeter of the cell. So they're not at the poles, they're all over, all around the perimeter, peritrichous. Here is another quick demo of the flagellate. This is of the flagellate stain technique. Incidentally, I tried to find a demo that would show you the stain as it was being performed, but all of the videos I found were demonstrating not only the staining technique, but also very, very poor safety practices. I mean, they're using bare hands and getting bacteria and dye all over them, and I don't want to use that as an example. So here is a good summary of the steps. And I did provide the link to the full video in the description because it includes more detail about how the staining procedure works.
The aim of the experiment is to stain the flagella of a cell by using Lifson's method. The requirements or the things we need for this staining method are as follows: flagellated cell culture slant, Lifson stain, 1% methylene blue solution, and distilled water. First of all, we have to take a slant of flagella cell culture and 2 drops of sterile distilled water. This sterile distilled water we will be adding by using a sterile pipette. After adding sterile distilled water, we have to incubate the slant for 20 minutes. Take a drop of suspension and add on a slide kept in slanting position. We have to air dry the smear and no heat fixation treatment is given to the smear. Flood the slide with Lifson stain and air dry the slide. After appearance of a shiny film, give a stream of water wash treatment to a slide. Treat the slide with 1% methylene blue for 1 minute. After that, wash the slide with water, air dry and observe under oil immersion.
Now for experiment 3, you will watch this video here that I made, and you'll practice identifying flagella. Remember that some of these are still going to be quite difficult to see, even with the stain and even with them being perfectly focused images. It still can be hard to see because the flagella are so thin and so slight that they can be viewed at a total magnification of 1,000 times the actual size, and then you can still barely see them sometimes. So be sure that you note the presence or absence of flagella, note the arrangements of the flagella that you see, and record your answer to the guided questions in the lab report 2 template. We didn't really have any safety guidelines for this lab since you're not doing hands-on experiments, but you should understand and know for the endospore and capsule stains, the safety stuff would be the same as it would be for any other stain with your acidic or basic dyes. And then for the flagellate stain, in addition to those safety regulations, you would have to use laminar flow because of the tannic acid and heavy metals. Now for your observations and interpretations, you want to make sure that you're able to differentiate vegetative cells, sporangia, and endospores in the images. Be sure you know the steps involved in germination and sporulation. Make sure you know how the endospore stain works. The capsules, you want to be able to differentiate the cell from the background in the capsule surrounding the cell. Make sure you understand how the stain works. Make sure you know why capsules don't take up the color of either of the stains, the acidic or the basic dye, right? And then for the flagellate stains, were you able to observe could you see them? Could you describe the flagella? And what kinds of arrangements did you see? Thank you guys for watching. Don't forget to do the reading. Check the description for more videos related to these topics, and leave your questions for me in the comments below.